Alright, welcome to the last part of the organelle notes. Please make sure that you have in front of you the learning targets for this unit. Make sure you are constantly checking those. Please also make sure that you have the note handout that should be linked on my website right underneath the link for this video. Okay, that way you don't have to write everything out. Okay, you guys did a really good job going through notes today and yesterday. I'd like to finish them up quickly so that we can move on. So my intent is for you to get through the rest of these quickly with the outline that I gave you, and then start focusing on 3.3 TIV, which is due on Thursday the 9th. Okay, so here we go. On the first slide, I will underline the words that you need to fill in, like this first one. Okay, we need to talk about the cytoskeleton. Cytoskeleton, it organizes organelles. Okay, so organelles is that first part you need to fill in. It provides structure, shape, and support, much like our skeleton will provide structure, shape, and support to our bodies. By the way, if we've already gone over this in class, I know a couple of my hours we got past this, go ahead and rewind until you see the last slide that you were on. Okay, now for those of you that this is where we stopped, you need to obviously take these notes. Okay, cytoskeleton also is going to help provide motility. Motility means its ability for some kind of movement. Okay, so cytoskeleton interacts with motor molecules. So if you remember on that Harvard video that we watched, that weird thing that looked like it was walking, okay, that's a motor protein, and it is quote-unquote walking along the cytoskeleton. Okay, so cytoskeleton, you can think of it as a skeleton, you can think of it as a highway or a road for these motor proteins to follow. Okay, cytoskeleton, oh, hold on, I didn't underline what you need to write from that last one. Interacting with motor molecules, make sure you have the proteins in there as well. Sorry. Next, transfers mechanical force into cells. So a mechanical force would be like a hit. Okay, a hit or drop or something like that. Cytoskeleton, we need to talk about structure. There are actually 3 different types of structure. If you have that handout in front of you, you can see right off the bat that those 3 types of structures are things like microtubules, the intermediate, and microfilaments. Okay, we need to go through all 3 of those. So let's get started. First one, microtubules are the largest out of all the cytoskeleton structures. Please note, however, that we're talking about microtubules. These are so super tiny. Okay, these microtubules are hollow tubes made of tubulin. Okay, so they're kind of like straws. Okay, I'm going to write that just in case that helps. They're like straws, not that they're sucking things in or out, just that they're these hollow tubes. Okay, microtubules can be assembled and disassembled. In the video you saw both the assembly and disassembly of microtubules. We will try to go back in class, we'll try to go back and watch that video again, or you can look it up. It's The Life of a Cell, I think. If you search on YouTube, you can find it. Next, they provide shape and support for the cell. Okay, and they act as tracks for organelles. Okay, organelles that have special motor proteins. They have motor proteins, it means they can move. Okay, it also will guide chromosomes during cell division. So if you remember, if you think back to learning about chromosomes and cell division, okay, you have these string things like this. Okay, well those are microtubules. Those microtubules then are going to pull those chromosomes apart. That step's called anaphase. Okay, I'm going to pull those tubes apart. That tube right there, that string, that is a microtubule. Okay, then microtubules can also be cilia, little hair-like structures that will help cells move. And then they can also be flagella. Okay, flagella are these little tail-like things that are also going to help cells to move. Well, bacteria, we're talking about, actually, that would be a good one on your table. If you had to have microtubules, microtubules can be found both in prokaryotic and eukaryotic cells. Okay. Next, we're going to talk about intermediate. I'm going to stop underlining because you should get the GIF and be able to do this no problem. So intermediate cytoskeleton is made up of various fibrous proteins that supercoil. Okay, they help reinforce cell shape, not shell shape, cell shape. Okay, and they anchor organelles in nucleus. So this is probably, if I had to say, this is probably the most important thing that you need to know. Okay, also know that they are permanent. Highlight those, circle them, something. Okay, they're about in between the microtubules and, oh geez, sorry, let's go back. In between microtubules and microfilaments in size. Leads us to our next one, microfilaments. This is the smallest of the cytoskeleton structures. It is made out of protein, just like your microtubules are. Okay, now take note of the fact that your intermediates are also made out of proteins, but they're made out of these fibrous proteins that supercoil. Your microfilaments are made out of actin, which is a protein, and they're twisted. You've got this double chain, and they are twisted. They can also be put together and broken apart, can be assembled or disassembled, just like your microtubules. And they form this 3D network that supports cell shape. Okay, so the others help with cell shape. This gives you that 3D network that's so important. Okay, they also maintain tension. Tension is a pulling force. And then with myosin, they're going to produce motion. They're pretty cool. Alright, so let's talk about cilia and flagella. First, flagellum. Okay, flagellum is going to be singular, flagella plural, just in case you're wondering. Okay, so flagellum gives you this whip-like action. So if you take a look over here, here we've got some flagellar movement. Okay, flagella, remember, are made out of microtubules. And you see how they're all just kind of circled around there. And it's going to move kind of back and forth like this. Whip-like action. Okay, cilia movement is going to be more of a sweeping action. I really can't show that to you with my pen. It just all is going to look the same. So go, if you want, look up, if you need more clarification, look up on YouTube, flagellum versus cilia. Okay, see what you can see. If not, ask me in class and we can talk a little more about it. Okay, so cilia has got the sweeping action. Sometimes cilia act as an antenna. Okay, they're non-motile, so the flagellum are going to help things move. Cilia are not. Okay, all vertebrate animals have primary cilia. It's used in embryonic development, sensory reception, and cell function. And we really still are learning a lot about it. We don't know that much. Okay, mechanism. Both cilia and flagella have got 9 microtubules attached to this basal body. So take a moment to look over here at the structure. Okay, so your basal body is here. It's attached down here to this basal body. Okay, and they've got 9 microtubules. So let's count and see. That's 1, 2, 3, 4, 5, 6, 7, 8, 9. Excellent. Okay, it is the same structure as centrioles, which we will get into later. And they use dinins to cause the cilia and the flagella to bend. Okay, so what are dinins? Well, for now, just know that they're these little arms that cause movement. Okay, we can get into that later. Because really, what I'd like to move into next is your learning check. Okay, on the handout that I've given you that is on my website, is this nice little table, and it says learning check, gives you the function, name of the organelle. Okay, so what I'd like you to do is to work looking through your notes and filling out that table as a learning check. Okay, so you'll need to pause it, flip through your notes, and fill that out. Okay, and then when you're done, come back and we'll talk about our very last target. Okay, so hopefully, welcome back, yay. This is your last target that we'll be covering in the notes, which is compare and contrast the structure of cells from different types of organisms. We already did that for prokaryotic and eukaryotic organisms. Okay, now we need to talk about plant versus animal. So, first one, Plasmodata, oh my gosh, sorry, end of the day. Plasmodesmata, okay, it is only found in plant cells. Okay, we're talking about plants. It's only found in plant cells, that's important. Okay, and what they are is they're channels through the cell wall and membrane. They're these little channels that allow for stuff to pass through. Allow cytoplasm to touch from the different cells. Like here, this is a great example right here. Plasmodesmata is this little channel here. It's going to allow the cytoplasm here and the cytoplasm here to exchange things like small molecules and H2O. Okay, that's Plasmodesmata. Think of it as little bridges between the cells. Okay, animal cells. Animal cells are a lot more complicated. I've only got a couple minutes, so I gotta go through this quickly. You'll probably have to pause it to get all the stuff. Okay, we have things called tight junctions. Tight junctions right here. They're like the thread. They're like thread that are kind of keeping membranes of other cells bound together very tightly. That's why it's called a tight junction. Okay. Cells, it says cells lightly pressed and linked. They're knitted together by proteins. You can think of them as leak-proof sheets. Anchoring junctions, these are called desmosomes. Okay, think of them as rivets. They're right here, or like a zipper. Okay, there's zippers, that are going to bind cells together. It helps to strengthen cells against mechanical stress, so like, I don't know, some kind of force, some kind of hit. Mechanical stress, think of your muscles. When you move your muscles, that's mechanical movement. So it helps those cells be strengthened by this interlocking here in between the desmosomes, or those anchoring junctions between cells. Okay, and then they're common in tissues that stretch. And the last one, real quick, are gap junctions. Gap junctions are similar to Plasmodesmata. Right here, it's that little channel that allows an exchange of ions and small molecules. I have like 10 seconds left, so pause it if you need to. That's it for our cells, for our slides, and I will see you in class tomorrow.